So this week we got respiration, last unit before the exam. Okay, we'll get into it. Feel free to come by to my office hours tomorrow if you have some last minute questions before the exam, 2:30 to 3:30. There's also my email, and then my notes. Feel free to use those too. Okay, so starting out, just looking at anatomy. So the respiratory system is divided into the upper respiratory system and the lower respiratory system. A lot of the upper respiratory system is the conducting parts, which just means that they're kind of parts where the air is just passing through to the parts where gas exchange occurs. So on the slide, the orange highlighted ones are the conducting zone, and then the yellow is the respiratory zone, I believe. So starting in the upper, you have your nose, mouth, pharynx. In your pharynx is just your throat, or anything superior to the vocal cords. And then the lower respiratory system, you have your trachea, which is the tube that brings the air in and out of your lungs. The trachea is lined with hyaline cartilage rings, so it has a rigid opening. And it's often confused with the esophagus. The esophagus is what brings food to your stomach in the digestive system. So that's separate from the trachea. Bronchi, there are 3 different types or branches of bronchi. You have the main bronchi, which divides into each lung. And then you have the secondary bronchi, which are also known as the lobar bronchi. So there's 1 secondary bronchi for each lobe of the lung. And then you have tertiary bronchi, which the secondary bronchi branch into. And then after bronchi, you reach the bronchioles. And after the bronchioles, you reach the alveoli, which are like the little grape-like structures at the outside of the lungs. And this is where gas exchange occurs. And those are surrounded by a lot of capillaries, so the blood could get really close to them, so gas exchange can go in and out of the blood easily. So here's what I touched upon on the last slide. The conducting zone are just kind of tunnels of air, or tunnels that allow air in and out of the respiratory zone. And the respiratory zone is where gas exchange occurs. And this includes the respiratory bronchioles too, and the alveolar duct and the alveoli. So here's just a visual. You can see the trachea dividing into the bronchi, which then divide into the bronchioles, into the alveoli. Something else to note is that the right lung has 3 lobes, while the left lung has 2 lobes. Does anyone know why the left lung only has 2 lobes? Yeah. Right. Yeah. So the left lung only has 2 lobes because of the heart, which means that it only has 2 secondary bronchi. The right lung has 3 lobes, so it has 3 secondary bronchi. And then here's just a visual for the top with like the nose, mouth, into the trachea. So you can reference that. So we just talked about this. The right lung has 3 lobes with 3 secondary bronchi. The left side has 2 lobes to accommodate for the heart. And then in the picture, you can see the secondary bronchi are highlighted in green. So you can see both of the lungs have 1 primary bronchi and then either 2 or 3 secondary bronchi. Okay, so now we're going to get into some histology of the conducting zone. So it's lined with epithelia, and the epithelia varies based on the location in the conducting zone. So the oropharynx and the laryngopharynx have stratified squamous epithelium, but most of the conducting zone has respiratory epithelium, which is pseudostratified columnar epithelium. And then the bronchioles have simple columnar epithelium, and then the terminal bronchioles have cuboidal cells. So you can think kind of as you get closer and closer to the alveoli, you're going to want to have more thin cells because that's where gas exchange is going to occur through the cells. So if you kind of look at the list, as you go down, you have stratified, which is like multiple layers of epithelium. Then in the respiratory section, which is most of the conducting zone, you have pseudostratified columnar epithelium, and then it gets down to cuboidal cells in the bronchioles. So as you move down, the amount of cartilage also changes. So in the trachea, we have a lot of cartilage, which enables the trachea to have kind of a rigid circular shape to allow a lot of air in. As you get down into the bronchi, the cartilage is broken up into plates. So there's some protection, but there's also some smooth muscle, which allows some give, or like some constriction and dilation. If you remember from one of the units before in the arteries had a lot of smooth muscle, which allowed us to either constrict or dilate. It's the same situation here. In the bronchioles, there's a thick layer of smooth muscle, which then allows us to either bronchoconstrict or bronchodilate based off of the amount of air that we need. So to allow that bronchoconstriction and bronchodilation, there's no cartilage and instead a thick layer of smooth muscle. So here's a visual of that. You can see the trachea has a thick layer of cartilage, and then the bronchi has some plates of cartilage with some smooth muscle, and then the bronchioles would have no cartilage and all smooth muscle. Okay, so now we're getting into the respiratory zone histology. So we have simple squamous epithelium that lines the capillaries. And then in the alveoli, there are two important types of cells. So you have type 1 alveolar cells, or pneumocytes. And these are simple squamous cells that do gas exchange with the capillaries. So these need to be in a moist environment or a watery environment to work well. And in this watery environment, it creates surface tension, which is a force that pulls molecules together, or it creates kind of a collapsing force in the alveoli. And this surface tension is due to the hydrogen bonds between water molecules in the alveoli. So basically, you have a bunch of alveoli, like say that look like that on the outer or perimeter of the lungs, and you have the type 1 cells, and the type 1 cells need to be in a watery environment, and these do hydrogen bonding between water molecules, which creates a force that pushes the alveoli in. It's kind of like a collapsing force. And this is important because when you exhale, you need the alveoli to kind of collapse to push air or carbon dioxide back out of your lungs to allow you to exhale. If your alveoli didn't collapse like this, if there's no force pulling in, then you wouldn't be able to get the air, you wouldn't be able to exhale. So when the alveoli collapses in, it pushes air up, back out, so you can exhale. And again, this is due to hydrogen bonding between water molecules, which is just like when you, which is creating that force flowing in. Type 2 cells, on the other hand, produce something called surfactant. And this is a chemical that lines the alveoli and prevents it from collapsing. So it's an opposing force in the opposite direction. And it does this basically by breaking the chemical bonds between water molecules. So if this force is due to type 1 cells, due to hydrogen bonding, then the opposite force due to surfactant, I'll do it. That doesn't mean it's a bigger force, it's just because we're talking about a different cell. And that's surfactant. So what surfactant does is it basically breaks the hydrogen bonds, which kind of allows the water to expand. And it pushes the alveoli out. So you have these two opposing forces that kind of maintains this equilibrium state almost of the alveoli being in the correct like pressure-volume relationship. Yeah, does that make sense? Does anyone have any questions on that? Okay. So surfactant also improves lung compliance. It allows the lungs to expand more by producing this outward force in the alveoli. And it prevents smaller alveoli from losing volume to larger alveoli. It prevents alveoli from collapsing and then needing to merge into one big one. And then there are two, yeah?
Sorry, it doesn't make exploration harder, right?
If you have too much, I think that's my next slide actually. Yeah, okay, we'll get there. And then there are two regions of surfactant, hydrophilic region and hydrophobic region. And they repel each other to keep the alveoli open.
Can you go over lung compliance one more time? Like, what does that mean?
Yeah, so by helping the alveoli kind of expand, it helps your lungs be able to expand and take in more air.
Okay.
Does that make sense?
Yes.
Okay. Okay. So smaller alveoli have less surfactant to make them more likely to collapse. So then they may lose air to larger alveoli, which reduces the surface area for gas exchange. So basically that's saying that a smaller alveoli has less space for surfactant, which therefore increases its likelihood of collapsing. But this says healthy people produce a little bit more surfactant in small alveoli to maintain gas distribution and maximize the surface area for gas exchange. So in a healthy person's smaller alveoli, there's a little bit more surfactant, which allows more air to get into those smaller alveoli sacs, and therefore more air to get into your lungs, more efficient gas exchange. And your question was if there's too much surfactant, does it make it harder to exhale?
Yeah, if the surfactant is like stopping the collapsing force of the alveoli, does it make it harder for them to like become smaller to expel the air out?
That would be a valid hypothesis, but I am not sure if exactly that could be a problem. But that would logically make sense. If there's too much surfactant, then it would be harder to exhale because it's making the alveoli larger. But yes. So here's a visual. You can see the surfactant has the hydrophobic and hydrophilic regions. And then, what else do you see in this? The surfactant is lining the inside of the alveoli. And then you can also see in this picture the fused basal lamina between the alveoli and the capillary. And that increases surface area. It makes sure that the capillary is completely touching the alveoli as much as it can because it's physically fused to it. So that increases the surface area for gas exchange, the fused basal lamina. So here's a summary of the two forces we just talked about. The two forces are opposing. The hydrogen bonding and surface tension from the type 1 cells pulls the alveoli in. And the surfactant disrupting the hydrogen bonds reduces pressure and maintains the shape of the alveoli by providing an outward force. And both of these forces are stronger in smaller alveoli, or have a greater influence. I know you guys touch on a bunch of respiratory diseases in this chapter, but here, this one directly relates. So emphysema is when the alveolar walls are degrading, which leads to larger air sacs that share the air. So that basically leads to a decrease in hydrogen bonding, a decrease in surface tension, and a decrease in recoil. So basically the type 1 cells with the hydrogen bonding and inward surface tension force is decreased. So the alveoli aren't recoiling as well. So that makes it hard to exhale. Okay, moving on to the mucus escalator in the trachea and pharynx. So here you have some goblet cells, which are mucus-creating cells. And these create a mucus layer that coats the trachea and pharynx. And this mucus traps pathogens in dust, and it also humidifies air as you inhale. And then there are cilia, which are kind of like little hairs that help beat the mucus or debris up out, so it can be expelled, so it doesn't get into your body. And the goblet cells are not in the bronchioles or the alveoli. Instead, in these regions, you have alveolar macrophages or dust cells, and these trap any pathogens that made it through the mucus escalator. Because if you had mucus in your alveoli, then you'd be disrupting gas exchange. So you don't want that there. Here's here's the respiratory membrane. It was on a few slides ago. This just describes the interface between the capillary and the alveoli in the lungs, the respiratory membrane. So then there's internal and external respiration. External respiration refers to the gas exchange from the lungs to the blood in the respiratory membrane. So that's between the alveoli and the capillary. Internal respiration refers to gas exchange in the tissues. So then the blood in the capillary takes the oxygen from your lungs, travels to your tissue, and then internal respiration refers to the tissue or muscle actually taking the oxygen from the blood and releasing carbon dioxide. And then we talked about before, the fused basal lamina increases the surface area of gas exchange on the respiratory membrane because they're actually like fused together. There are no spaces between. So gas exchange can occur at like every point along the membrane. Getting into some more external respiration, gas exchange. So air crosses the simple squamous cell, the type 1 cell right here in the alveoli, crosses a basement membrane, and a second simple squamous cell, or the endothelium cell in the capillary to enter the blood. So basically, gas is passing through type 1 cell, there's the fused basal membrane, and then right here is a second cell of the capillary into, this is the blood. So then carbon dioxide and oxygen, they're both lipid soluble and readily diffuse through the membrane. However, carbon dioxide is 24 more times soluble than oxygen. So carbon dioxide can rapidly diffuse through the tissue and dissolve in the plasma. So this is why chemoreceptors sense changes in carbon dioxide rather than oxygen in the body. So changes in carbon dioxide can be rapidly detected, and breathing can then be changed to adjust based off of concentrations of carbon dioxide. And 7% of carbon dioxide dissolves in the plasma. 2% of oxygen dissolves in the plasma because oxygen is more nonpolar than carbon dioxide. So less nonpolar oxygen can dissolve in the polar water. And then 98% of oxygen binds to hemoglobin to be transported throughout the body. So then here's a little bit more of like the chemistry of how this is actually working. If you remember, like dissolves like, so blood is mostly water, which is a polar molecule. Carbon dioxide combines with water. We'll go over this more later, but here's just like a summary. Carbon dioxide combines with water to become carbonic acid, which is polar. Carbon dioxide dissolves in the blood in the form of carbonic acid. So it forms carbonic acid so it can dissolve in the blood. And then again, the chemoreceptors detect the amount of carbon dioxide in the blood because it's dissolved actually in the blood. Whereas oxygen is nonpolar and must bind to hemoglobin to be transported. So there are chemoreceptors to detect the amount of carbon dioxide because there's more carbon dioxide actually dissolves in the blood plasma because it can, because it's more polar. So where carbon dioxide goes in the blood, So blood acts as a buffer, and we'll see this equation a few times. So carbon dioxide readily dissolves in red blood cells and combines with water to form carbonic acid. 93% dissolves in the red blood cell. 23% of that 93% binds to hemoglobin to form carmamide hemoglobin, whereas 70% combines with water to form carbonic acid. And then 7% remains in the plasma, dissolved as CO2. Okay, so here's the equation broken down a little bit more. Carbonic anhydrase is the enzyme that combines carbon dioxide and water to form carbonic acid, which is H2CO3 in the middle. And then carbonic acid, once it's formed, readily just dissociates into hydrogen ions in the bicarbonate ion, which is HCO3. So when carbonic acid readily dissociates, we're releasing hydrogen ions, which increases acidity. So on the right side of the equation, where we have more acidic conditions, we have a lower pH. However, hemoglobin can carry this H plus and buffer it to help increase the pH or make it more basic, so then we can drive the equation further to the right, if that makes sense. So HB comes and picks up those H's, so then we're increasing pH and can keep forming more carbonic acid when we have a lot of carbon dioxide to use it up. And then bicarbonate also needs to be gotten rid of to keep the reaction moving right. We got rid of some of the H. So then there's something called a chloride shift, which acts as an antiporter to exchange bicarbonate for chloride to keep the reaction being pushed to the right. Okay, so I kind of want to go to this slide because this helps me visualize it better. So you can see in the pink, carbonic anhydrase is that enzyme that combines carbon dioxide and water into carbonic acid. And then carbonic acid, once it's formed, readily dissociates into the hydrogen ion in bicarbonate. Hemoglobin picks up some of that hydrogen ion to buffer the blood, forming HB, which increases the pH, makes it more basic by taking some of the H out. And then bicarbonate is able to be exchanged for chloride with the chloride shift to help the equation be driven to the right. So you can think, if there's more carbon dioxide on the left, hyperventilation is increasing your breathing rate. So increasing your breathing rate will increase the amount of carbon dioxide in the body. Okay, no, I said that wrong. Hyperventilation will decrease the amount of carbon dioxide in the body because you're expelling more carbon dioxide, which then pushes the equation left because it's reversible. So you can think of it as if you're losing something, this is the way I think of it in my head. If you're losing something on this side, so if you're breathing out too much carbon dioxide, then the equation is going to want to automatically go this way to make it equilibrium. Whereas on the right, hypoventilation, breathing more slowly, you're going to build up more carbon dioxide in the body, which is then going to push the equation towards the right. So you have more orange carbon dioxide, the reaction is going to run pretty quickly forward. And naturally that leads to respiratory acidosis. So we're forming more hydrogen ions. And then on the other side, it leads to respiratory alkalosis. Okay, so we'll back up a little bit. So this is what we just talked about, but more broken down into words. So a left shift is done by hyperventilation. Or hypocapnia, which is decrease in CO2. So you're breathing out more CO2. So if you have less CO2, then you're going to do a left shift because you're cutting out something that you need on that side of the equation, so it's going to move towards the left. And that leads to respiratory alkalosis, which leads to a higher pH. And to treat this, you can have someone breathe into a paper bag to increase the amount of CO2 that they're breathing in, to try to help move the equation more towards the right. And then a right shift is caused by more carbon dioxide, and this is done by hypoventilation or breathing slowly, or hypercapnia, which is more carbon dioxide out. Or no, more carbon dioxide due to not breathing enough carbon dioxide out. And this leads to respiratory acidosis. I suggest drawing something like this by on your own. It really helps visualize the equation. It makes it make more sense. So here's just some vocab between ventilation and perfusion. Ventilation is just gas coming in the lungs and reaching the alveoli, whereas perfusion is the blood flow in the capillaries of the alveoli. So ventilation and perfusion should be matched in a healthy individual. Areas with increased ventilation should have good perfusion to maximize gas exchange. And then this can be adjusted by local control or by nervous system control, by bronchodilating or constricting areas that are better ventilated in the lungs. And then you guys looked at these values a lot in the lab, I believe. So these just describe different volumes of air in your lungs. And I have the definitions here. So keeping the ventilation terms is kind of tricky. So there's the dead space, which is areas where gas exchange does not occur at all in the lungs. And then the tidal volume describes the small amount of air that flows in and out with a normal breath, which is about half a liter of air. Inspiratory reserve volume is the volume of air that can be inhaled beyond the tidal volume. So that would be forcing an inhale after a normal inhale. Expiratory reserve volume is the volume of air that can be exhaled beyond normal tidal volume. So after a normal breath, that would be pushing out as much air as you can. And the volume of air that comes out is the expiratory reserve volume. Vital capacity is the total volume of air that can be exchanged in and out. So it's the inspiratory reserve volume plus the expiratory reserve volume plus the tidal volume. So basically, a normal breath plus the amount of air that you can force in plus the amount of air that you can force out. And that does not include dead space. Total lung capacity is the amount of air that fits in the lungs, including the dead space. So total, you're including everything. So that's the tidal volume plus the inspiratory reserve volume. And you can never get rid of all the air in the lungs. You always have to have some dead space or dead air in the conducting zone. Anatomical dead spaces describe air being occupied in the non-respiratory part of the respiratory system. So air that's just kind of chilling in like the trachea, for example. Like there's always air there, but it's not doing gas exchange. So it's known as anatomical dead space. And then residual volume is the air left in the alveoli that cannot be exchanged. Physiological dead space is failure of the respiratory zone to do gas exchange. So that would be in like a damaged alveoli in emphysema. It's physiological dead space. I'm not going to go through all these, but I basically just compiled a list of all the different diseases that you should be familiar with by the end of this chapter specifically, just because I think it's easier to look at them because you can get kind of confused with like unfamiliar new terms. And then I did not go through gas laws or nervous system regulation, just so you know when you look at these slides when you're studying, just be sure to review them. And then I have some top hat questions. How many lobes does the left lung have? Secondary bronchi. Okay, I'll go back to this one. Nice. 2 lobes, 2 secondary bronchi. Right has 3 lobes, 3 secondary bronchi. Rank the following conducting zone tissues from most to least cartilage. So most cartilage, trachea, second most bronchi, and then bronchioles. Water molecules on the type 1 alveolar cells create what force on the alveoli? Perfect. Surface tension that pulls the water molecules together via hydrogen bonding, which creates a collapsing inward force on the alveoli. Type 2 cells produce surfactant, which produces an outward force. Oh. Okay. What direction is the force that surfactant creates on the alveoli? What does it do to create this force? What force does it oppose? Nice. All these look good. So it produces an outward force by breaking hydrogen bonds, and it opposes hydrogen bonding or surface tension created by type 1 cells. What is a pneumothorax? I didn't go over this, but thought I'd throw it on there. Yeah, so I don't think I put the correct answer in, but that is correct. So some of you emailed me about some study tips, and something else you can do if you're looking for some more practice is a lot of the times when questions are made, the responses could be a potential answer to another question. So you could go through this question and be like, oh, okay, what disease is chronically narrowed conducting zone? Is a chronically narrowed conducting zone? And then go through your notes and make sure you can answer the question with different variations. Because oftentimes on the exam, the questions do come from similar questions made and presented in class. Combining chloride with bicarbonate causes what? You guys got it, it's a right shift. So just to bring this back up, if you're decreasing a product on the right side, then the reaction is going to want to go that way to make more of the product. So taking away some bicarbonate would then make the reaction go towards the right. What does hemoglobin do to buffer blood? Okay, we're going to edit number B to say increase blood pH, or letter B. So hemoglobin does accept a hydrogen ion, but it does it to increase blood pH to move the reaction towards the right. So we're going to edit this. So by accepting a hydrogen ion, it makes the blood more basic. Which leads to a right shift. Select 2. What did you say?
It only allows you to select one.
Oh, okay. Yeah. So here are the 2 right answers: hypercapnia and hypoventilation. Hypercapnia is increased carbon dioxide due to not breathing enough carbon dioxide out. So hyper is higher, and then capnia, carbon dioxide. Higher carbon dioxide is going to lead to a right shift. Hypocapnia, decreased carbon dioxide, is then going to lead to a left shift. I think that's all. That's all I have for you guys. Feel free to stick around, ask me some questions, or come to Mouth Stars tomorrow, or email me.